Vancomycin has been called the antibiotic of last resort for decades. The drug doctors reach for when every other option has already failed. But a growing number of hospital-acquired infections, caused by a bacterium called Enterococcus faecium, have evolved resistance to it, turning what used to be a reliable backstop into a coin flip. On June 16 this year, researchers from Cold Spring Harbor Laboratory and Scripps Research, publishing in Nature Communications, reported a way to make vancomycin work against these resistant bacteria again. Instead of inventing a brand new antibiotic from scratch, a process that typically takes over a decade, the team found a small molecule that disables a bacterial enzyme the resistant strain relies on. And when they combined that molecule with ordinary vancomycin in the lab, the antibiotic regained its ability to kill the bacteria outright. Let us sort the signal from the noise. Understand why one drug's declining effectiveness is worth this much attention. Vancomycin-resistant Enterococcus, commonly abbreviated VRE, is not some rare laboratory curiosity. It is a real and rising cause of bloodstream infections in hospitals, particularly among patients who are already immunocompromised or recovering from major surgery. Enterococcus faecium becomes resistant largely by altering the structure of its cell walls so that vancomycin can no longer latch onto it and do its job. Once that resistance takes hold, doctors are pushed toward second or third choice antibiotics, drugs that are often less effective, more toxic to the patient, or both. The World Health Organization has flagged vancomycin-resistant organisms as a priority concern in the broader antibiotic resistance crisis, a slow-moving problem where bacteria are evolving faster than the pharmaceutical industry is replacing the drugs they defeat. A genuine way to restore an existing, well-understood antibiotic rather than waiting years for a brand new one addresses that gap directly. Not just one hospital's infection numbers. It is worth explaining exactly how the fix works, because it is clever rather than complicated. The resistant bacteria rely on an enzyme called secreted antigen A, or SagA, which helps remodel their cell wall during division. That remodeling process happens to be part of what lets the bacteria root around vancomycin's usual attack. The Cold Spring Harbor and Scripps team identified a small molecule, named PGHI4, that blocks SagA specifically. On its own, PGHI4 does nothing to kill the bacteria. Combined with vancomycin, it disrupts the cell wall remodeling the bacteria depend on. And that reopens the door vancomycin needs to do its job. This approach is called an adjuvant strategy, pairing a helper molecule with an existing drug rather than replacing the drug outright. And it has real precedent. The antibiotic combination of amoxicillin and clavulanic acid works the same way, reviving an older drug by disabling the enzyme bacteria used to resist it. The researchers are already developing more potent second-generation versions of PGHI4. Consider the economics behind why this approach matters as much as the chemistry. Developing a genuinely new antibiotic from scratch typically takes more than 10 years and well over $1 billion. And even after all that investment, many antibiotics generate relatively thin profits because doctors deliberately use them sparingly to slow down future resistance. That combination has pushed several major pharmaceutical companies to scale back antibiotic research entirely over the past 2 decades. Even as resistant infections keep rising, an adjuvant molecule like PGHI4 sidesteps much of that timeline, because it works alongside a drug, vancomycin, that regulators, doctors, and hospital pharmacies already know inside and out. Its safety profile and dosing are already established. That does not eliminate the work of testing the new combination therapy properly. But it is a meaningfully shorter and cheaper path than starting an antibiotic from zero. Which is exactly why researchers increasingly see reviving old drugs as one of the more realistic strategies for keeping pace with resistant bacteria. A real caveat belongs here. This result comes from laboratory bacterial cultures, not an animal model and not a human clinical trial. And the research describing it does not report either of those next steps having happened yet. The combination was tested specifically against Enterococcus faecium, one resistant species among many the world is currently contending with. So it is not yet clear how well the same SagA blocking approach would work against other resistant bacteria without further dedicated research. Laboratory discoveries at this stage have a well-documented tendency to stall out. Drug development has multiple gates. Safety in animals, safety in humans, effectiveness at real-world doses. And most early discoveries never make it through all of them to an actual prescription. The researchers themselves describe this as a foundation for developing more potent second-generation versions of the molecule. Language that signals promising early science, not an imminent clinical treatment. The chemistry and the mechanism are genuinely solid. Peer-reviewed work. The distance to a hospital pharmacy shelf is still substantial. Quick recap. On June 16, researchers from Cold Spring Harbor Laboratory and Scripps Research, publishing in Nature Communications, showed that a small molecule called PGHI4, which disables a bacterial enzyme named SagA, restores vancomycin's ability to kill drug-resistant Enterococcus faecium in the lab. So, signal or noise? Signal. The mechanism is real, peer-reviewed, and represents a genuinely promising adjuvant strategy for fighting a rising category of hospital infections without waiting a decade for a brand new antibiotic. Noise risk shows up if you read this as a finished treatment. This is cell culture stage research, with no animal or human trial results reported yet. Tested against one resistant species so far. The honest takeaway: a real scientific win worth tracking as it moves toward animal studies and beyond. Not a cure sitting on a pharmacy shelf next year. Subscribe for the health stories that separate a lab breakthrough from a finished treatment. Signal from the noise. Every week.